So we're going to talk today about the topic of senolytics. We have a special guest. She's talking all the way from Germany. It's Dr. Tharma Palin. She has just published a paper, February of this year, entitled "Senolytic Compounds Reduce Epigenetic Age of Blood Samples in Vitro." Thank you so much. I wanted to ask you. First of all, just introduce yourself and this paper, basically how it was done. I'm intrigued to know how you did this test on blood. So anyway, thanks very much for talking to us.
Yeah, thank you for having me. This is great pleasure to talk my results, and thank you, Gajraj, for having me. I come from and I originally come from Sri Lanka, but I'm studying in Germany. I finished my PhD in Aachen, Germany. It's the university called RWTH, Institute of Stem Cell Biology in Professor Wolfgang Wagner's group. The group is mainly interested in DNA methylation, epigenetic clocks, senescence, aging, and in mouse and human, and also some hematology malignancies.
Good.
My PhD mostly in the field of DNA methylation changes and aging clocks in the hematology malignancy, and also some drug treatment in such one paper as senolytic drugs.
Excellent. Well, yes, if you could tell us about this study, how it's performed, and what your results were.
So when we started to do this experiment, our question was, is there a senescent cells in healthy humans? Because in our previous study, we found in the hematological malignancy, there is a presence of senescent cells. Then we wondered that people always shown that senescents do accumulate with aging. And if we can detect this in the healthy blood samples without any history of a disease, for that we collected the blood sample in the age range of 20 to 85, and then we categorize somehow the old and young in different ranges, and then we just did a senescent staining. So one is like the beta gal, which is a prominent senescent staining, and also we can stain them using a C12 FTG antibody. This is like a cell surface marker, where it's classically used to detect the senescent cells, where we shown so that there was a accumulated senescent cells increase with age. And this gave us an idea, okay, why don't we treat them with the senolytic drug just in the cell culture plates and see what's happening? Because most of the studies in mouse or in cell culture, the senescents are artificially induced. They are rather by irradiation of a UV or some small chemicals by some drugs. They induce senescence, and they see how the effect of senolytics. But what we thought is like in human body, the accumulation senescence, rather than artificially inducing them, just looking at the presence of senescent cells and then see how the treatment works.
How many blood samples did you take?
In total of 18 to 21.
How are you looking for senescent cells? Were you able to detect them?
Yeah, I can show you maybe a couple of my slides because this nicely have some images, might be quite interesting. So this is the paper that we published, and then so this is the results that I was mentioning, that with increasing age, with the senescent staining, we could see an accumulation of senescence. There's a tendency, which is increasing with the age. And then what we looked here is like when we isolate some PBMC, so those are like peripheral blood mononuclear cells that you can isolate by taking a blood and do a FICO gradient, and you only take the mononuclear cell which also have the stem cells inside. And then we looked it up under microscope, and where you with the senescent staining, where you could see this prominent big cells, which is a morphology of senescence, which is already known, that these cells are already present in the old blood samples. And then when we did the treatment, we could see that we could clearly able to clear them by one of the drug treatment that we had. So this is like done by looking at the microscope. Then we further also looked at some quantification by doing some flow cytometry analysis for the C12 staining. Here you could see that untreated versus treated, we could clearly eliminate, this was around 12% of senescent cells. We could make them under 1% by this drug treatment. So this was for 3-day treatment. We cultured the PBMCs, the blood cells, and we put the drugs inside. And after 3 days, we took the cells and did the surface markers analysis. And we had like 8 different drugs in our study because senescents have very different mechanisms to activate them into senescent cells. And also they are involved in different pathways which are interfering the senescent cell to resist to apoptosis. So we picked the senolytic drug which is in the literature. Either they are already been in a clinical trials, or they are in the mouse studies, or they are just done in the cell cultures, but they are rather interfering in different apoptotic proteins. Then we could see that some of the senolytic drug could significantly reduce the amount of senescent cells when we compare to the untreated ones. And the P16 we did by gene expression profiles by using qPCR. It is one of the classical senescence marker where we could also see there's a downregulation of the gene after the senescent treatment.
Excellent. So I didn't know there were senescent cells that you could look at in the blood. So what about giving blood transfusions? Is that, I mean, does that put more senescent cells into the donor?
So I would say that this blood transfusion of young and old mouse, mainly this I forgot the name, but like you do a blood transfusion from the young mouse to an old mouse, and it has been shown by different immune compounds what's inside this regenerate the old mouse to make it younger, functionally younger, and also increase the lifespan. So I think this could be one of the reason. I think the main reason is you have better immune cells in the young body rather than the old. And also there's the presence of senescent cells might be lower in the younger. So when you do the transfusion, then this can be effective.
I see on that y-axis you've got D plus Q. Both those compounds are now available. Could you tell us a little bit about those? And perhaps do you think people should be on Q-certin?
So this D plus Q is this one of the main studies which found on around 2018 this dasatinib, which is a tyrosine kinase inhibitor, and this quercetin. Not sure how to exactly pronounce this, and this is also come from a very naturally occurring flavonoid, and people have shown that this senolytic cocktail in mouse have been reducing the pro-inflammatory cytokines in mouse and also increasing the lifespan of this mouse. So we thought this might be effective in our study, but rather I would not say that this didn't give a significant decrease in our study.
Excellent. And so maybe we could finish up. What is your conclusion from this study?
So one of our idea was like, so our group was interested in the epigenetic clocks, so to see if there is a difference between epigenetic age by treating them, because now we see that healthy cells with age do have senescent cells, and treating them with senolytic do reduce the senescent cells. And then what is then happening to the epigenetic age of those samples? For that, we have some kind of a targeted analysis for certain. I can go in detail if you want to explain how an epigenetic clock works, but in a very simple term, this is like a biological age or epigenetic age of a patient, of a person which is different from the calendaric age. And if you are in a calendar age, it's 28, but due to a lifestyle or family history or just some treatments, or stress level, anything, this can change your biological age. And people have shown that DNA methylation can be used to predict this epigenetic age. And we have utilized the epigenetic clock here. By after the treatment, what we see is that we could able to reduce the age of the sample, and this reduction was like surprisingly not depending on the age. We see the reduction independent if it's come from 20 or 80 years. Thus in the cell culture plates, these cells do have senescence, and they could able to regenerate or remove the old cells and make them younger more than around 5 to 10 years. But still we are not definitive about it because it's only 18 to 20 samples, and also the concentration of the drug will be depending on the different patients. So here we are more suggesting that we can use epigenetic age as a tool to understand the treatment strategies, because senescent cells do vary, and also to see if a drug treatment worked or not. They don't have a very prominent markers available to understand this, and we suggest here maybe we could use epigenetic clocks in addition to understand the drug treatment and its significance.
Good. So we have a biological age
Yeah.
and we also have a chronological age. So I think this is an extremely interesting paper, um, really about senescent cells and decreasing senescent cells, which may decrease DNA methylation.
Mhm.
DNA methylation is an indicator of biological age. Would that be a reasonable thing to say?
I don't know if I could say it will decrease the DNA methylation, but I would say that the clocks that we modeled have shown very good linear or non-linear correlation with age. So some of the CPG sites, so we could say like some of the regions in the gene which like adds a methylation or remove a methylation, which have a very good correlation with age. And people have also showed this is conserved between mammals. In the whole, the mammals can have a similar epigenetic, similar CPGs which are conserved. So we can utilize this to understand if these regions have been a significant increase or decrease in methylation. It can be both, depending on the CPGs that we pick. And by looking at those CPGs, we can make like a simple signatures to predict the epigenetic age. So I would say Howard came up with the very big CPG models, and in our group, we are trying to also in before we did like 3 CPGs or 9 CPGs by only looking at few CPGs as a targeted assay, which is also can simplify the clinical usage.
Very good. So what we like to do in the study of anti-aging is to look at biological markers. And I'm going to put up a list of biological markers, but I would think that looking at senescent cells would be a good biological marker. Would you agree with that?
Yeah, agree, definitely. It's one of the hallmarks of aging and also in cancer, interestingly. So there are interlinks between cancer and aging, and one main is cellular senescence, which have been shown. And I think senescence is very interesting phenomena. Like it has been used to been known to be helpful in patients like tissue remodeling and tissue repair, and also in the other way, the kind of cytokines or the pro-inflammatory chemicals it produce, it can also be very detrimental to. So it's such an interesting concept.
Yes, your paper is excellent. I do review every day publications that would be of interest to me, and yours came up this month, and I do really appreciate for you, coming and discussing your paper. I think it's very elegant and very extremely interesting, and I think anybody who's interested in the science of anti-aging should definitely read this paper. So I want to thank you again,
Mhm.
and I wish you the best in your career. I understand that you are at the beginning of your career as a scientist, so I do wish you the very best of luck.
Thank you for the opportunity. It's really great to present the work to general public because that's how the communication of the science should be, and it's a really great opportunity. Thank you for having me.
Okay, thank you.